Sorry, got more people in the waiting room. Just get ready to start. Just getting everything up. Where is it? Okay. Let's get going. Okay, so today we're going to talk about gas exchange. We have talked about kind of basic kind of exchange principles in the last lesson when we talked about transporting animals. We talked about the need for a short diffusion distance, the need for a large diffusion area, and we talked about the need for a close blood supply. And these are kind of things that will be reiterated throughout this kind of talk. So today we're going to be talking about kind of adaptations of gas exchange across the body of a single-celled organism, adaptations of gas exchange in insects, in fish, and in plants. And we're going to slowly work our way through gas exchange of different organisms. So how does gas exchange work in humans? Let's start with humans, actually. So gas exchange in humans is carried out by the respiratory system, which kind of makes sense. So the respiratory system is kind of all the way from your nose, through your mouth, down your pharynx, down your trachea, to your bronchi, which then lead into your bronchioles, and then into your alveoli in the other parts of your lung. So oxygen comes in to the lung, and it comes into the lung via the mouth or the nose. It then passes out. It then passes into the bloodstream. In the same vein, carbon dioxide, which is produced by actively respiring muscle cells, is in the bloodstream. It comes through the blood back to the lungs, where it is diffused out of the lungs, back into the alveoli, back up and breathed out. So there's a bit of an exchange here. Oxygen goes down its concentration gradient and goes into the blood, and carbon dioxide also goes down its concentration gradient from the blood into the alveoli. And then there's exchange. And this is kind of your general structure. So let's label this anatomy. We've got a bit of anatomy here. So can anyone name what this is? What this structure here is? It's kind of a cartilaginous bit. That's not the trachea. This is the trachea. So the larynx will be the tube behind this. This is something called the thyroid cartilage, which is a piece of cartilage that exists kind of above your thyroid, and you can see kind of it's in that position. Can anyone name this structure? Put it in the chat if you can. Take a second. No, that's not your voice box. Your voice box is actually a series of It's a series of fibers that run between your thyroid cartilage and this other structure. This is your hyoid bone. So someone said cricoid. It's not your cricoid. This is your hyoid bone, which again, it forms part of that respiratory system because your hyoid moves down as you breathe in. And you can see that because your Adam's apple is this, for men anyway. It's the protruding bit of your thyroid cartilage. It protrudes out. And as you can see, when you breathe in, that moves down. Your trachea exists here, which then splits into your left and right bronchi. Your bronchi then split into your bronchioles. And at the end of your bronchioles, there is your alveoli, which is kind of shown down here. I've been asked a question, good question. Do I need to know the hyoid bone for the exam? Probably not, no. It doesn't come up on any spec. It's just it's important just to orientate you when you're looking at where the pharynx ends. So the larynx is from your nose all the way down to the bottom of your mouth, which is your oropharynx. Your oropharynx runs from your pharynx to your hyoid. So your hyoid will be an important anatomical landmark for you to know where the pharynx ends and the trachea begins. Is this applicable to the OCRA exam board? Yeah, all of this comes from OCRA. But your simplified anatomy is kind of, you imagine your larynx exists behind this bone, because again, your larynx is a tube. Then your trachea after this kind of structure, your bronchi, your bronchioles, and your alveoli. That's the main anatomy you need to know for your exams. And that's all you'd be expected to know. So air enters the lungs through the mouth and nose into the larynx. Air from the larynx passes into the trachea. The bronchi, the trachea split into two bronchi, left and right bronchi, and each main bronchus splits into lots and lots of smaller bronchioles. Your alveoli are at the end of the small kind of bronchi and are specialized for gas exchange. So let's talk about this gas exchange pathway. So air can pass into the pharyngeal cavities in two ways. It can pass into the pharynx either through the nose, your nasopharynx, or through the mouth, which is your oropharynx. And this is really important to note the difference between these, because this is important structurally because it determines the humidification of air. So one of the things that you do need to know for your exam is, and this comes up in all the specs, is air is humidified as it enters the nose. It is not humidified as it enters the mouth. And humidifying air basically means you add water vapor to it. This adding of water vapor basically steepens your concentration gradient, which makes the gas exchange more efficient, which is why you're encouraged when you breathe in to breathe in through your nose, and when you breathe out to breathe out through your mouth. It's just the way you're kind of, that's the encouragement to do, and that's because of the humidification of air that happens in the nose. Yeah, so the air from the nose and mouth, nasopharynx, oropharynx, and then into your larynx. And atmospheric air can be dry, and to overcome this, air is humidified in the pharyngeal passages. The nasal pharynx, the nasal passage, is also lined with ciliated cells, so columnar ciliated cells. So you've got goblet cells and you've got cilia. The goblet cells produce mucus. The cilia helps to waft away the aspects that are caught on the mucus. And again, this helps filter out blood, dust, bacteria, and viruses from the air. Mucous membranes in the nasal and oral passages help in humidification of inspired air. and to help trap dust and bacteria. Now we're in the trachea. So the trachea is a cartilaginous tube. Now, if we think about a similar structure in plants that is also cartilaginously lined or lined with thickening, it's also thickened, we can think about the xylem. And why would, and we talk about xylem being lined with lignin, and why is the xylem lined with lignin? Yeah, right, is the xylem. Why is that? Why is the xylem lined with lignin? Yeah, to reduce the evaporation of water, but it is also for a second purpose. Support, yeah, big structural support. The xylem is, the water in the xylem is under very high pressure. So if we think about, if we parallel that back to our bodies, and this is what, again, is a bit of a revision tool. Parallels, making parallels between structures is a great way of kind of thinking about it. And often you can get really random questions where they ask you to compare the xylem and compare the trachea in exams. So making parallels and drawing parallels between the structures of different beings or different organisms can be a good way of remembering things. And especially when we talk about insects and we talk about fish, this comes in handy a lot more. But yeah, so your trachea is lined to basically, because the air is at really high pressure in the trachea. So in order to prevent your airways from collapsing, you have the high pressure in the trachea. The tracheal cartilage are specialized cartilaginous rings in the tracheal walls, which help prevent the trachea from collapsing. The goblet cells, again, these are cells that produce mucus. And again, that job is to trap dust and particles to prevent bacteria and organisms from entering the lungs, which would be quite bad. There's also ciliated cells, which are lined in the trachea, and they help trap dust and pathogens. Now, from your trachea, you have your bronchus and your bronchioles. So your main bronchi still has the ciliated epithelium or the ciliated cells to again help waft mucus and other pathogens away. And your bronchi are also still supported by cartilaginous rings because, again, the air in the bronchi is still at very high pressures, which means that they are still at risk of collapse. And each bronchi splits into many bronchioles. And I've been asked a question in the chat. Will I be doing a summary whiteboard at the end? Of course, I'll be doing a summary whiteboard, yeah. Bronchioles are small, fine extensions of the bronchi, which branch off into different regions of the lung. This is called a bronchial tree. And bronchioles have, again, ciliated epithelium cells to filter the air of dust and pathogens, but also smooth muscle, because this is for bronchoconstriction and bronchodilation, which is an important mechanism for protecting your lungs. Because remember, if you have air, if you have allergens or anything that could be potentially toxic to your body does not want that entering through your air. So it will constrict your bronchi, and it will basically prevent that from entering the alveoli. And again, this can occur in response to various stimuli from the nervous systems. And I'm not really going to talk about bronchodilation and bronchoconstriction because, again, that's more of an A2 concept, talking about the neurotransmitters and the hormones that do that. Alveoli are the primary surface for gas exchange in humans and most vertebrate organisms. They are small sacs and found in clusters at the end of bronchioles. Air enters the alveoli through the bronchioles. The entry of air inflates the alveolar sacs. This inflation increases their surface area. And again, the alveoli are made of elastic fibers. These elastic fibers help to stretch and recoil, which will help control their shape, but also help push air in and out of the cells, of the alveoli. alveoli, sorry, I've been asked a couple of questions. Surely the C-shaped cartilage rings help prevent the trachea from collapsing? Yeah, they do. So that's the idea. The cartilage is there to help prevent the trachea from collapsing because the air is at really high pressure. What does inflate mean? So inflate means to increase in size. So when you inflate a balloon and you blow up a balloon, you're inflating the balloon. You're increasing the surface area. How can smooth muscles constrict and dilate in bronchioles when it has cartilage? Surely cartilage is rigid. Yes, cartilage is rigid. Well, not fully rigid, but yes, cartilage is kind of rigid. So I'll picture it for you. I'll draw you out a picture of it. So at the points of the cartilage, while you are completely correct that those can't dilate or constrict, between the cartilage, you can have incredible dilation, which still overall narrows airflow. Because you've dilated, you've constricted between, which means that the pressure in between is still quite high. And so although cartilage is rigid, while you were right, in between the cartilaginous rings, there can still be some bronchoconstriction and bronchodilation. Pulmonary surfactant. So pulmonary surfactant is a fluid that helps prevent alveoli from collapsing. Basically, it does that by kind of maintaining the pressure so that the alveoli are never empty, so it prevents them from collapsing. And it also helps keep the alveoli dry. Alveolar wall is only 1 cell thick. It's very thin. And this thin wall makes diffusion of gases more efficient because it makes it more efficient for gases to diffuse across. The alveoli are wrapped in capillaries. Alveolar capillaries carry deoxygenated blood from the heart to the alveoli, whereas CO2 diffuses out of the blood and O2 from the alveoli diffuses into the blood. There is a high surface area. So alveoli are designed to have a high surface area. And again, this increases the rate of diffusion and the efficiency for gas exchange. Other kind of features of alveoli. So let's go to a summary whiteboard at this point because I think this would be quite useful. So I will put up a summary whiteboard. And I'll quickly, so these are kind of your important notes to take down. Ooh. Check, do I have these notes? Pulmonary surfactant is kind of like a fluid. And this fluid is really important for keeping the alveoli. Basically, what it does is it coats the alveoli and it traps any other fluid that's trying to stick onto the surface of the alveoli, which basically means that the alveoli stay dry throughout. Sorry, I'm just getting the notes up. Do we need to know this in the exam? Yes, you do. You will be expected to know this in the exam. All right, it appears that I've lost my notes from my A-level, so I don't have a summary whiteboard to give you for this topic. So I've got all the other ones for the other topics, but for this topic, apparently I don't have my notes anymore from when I did this. So fortunately, I don't have this summary whiteboards to give you for this. I can give you the processes, but I won't be able to summarize kind of just the general anatomy side of it. So deoxygenated blood from the heart travels to the lungs. And so remember, we talked about this last session. We talked about it traveling from the pulmonary artery to the lungs. And these splits into loads and loads of capillaries, and each of these capillaries wrap around a single alveolus. CO2 diffuses out of the blood and into the alveoli. the newly oxygenated blood now travels through the pulmonary vein back to the heart, where it then travels out of the aorta and to systemic circulation. Okay, we'll talk about this because this is really important. It's ventilation. So movement of air into and out of the thoracic cavity. So the thorax or chest is made up of a rib cage, which houses the heart and the lungs. Remember, during breathing, so inhalation and in exhalation, the thoracic cavity expands and contracts. Expansion and contraction allows for changes in air pressure from breathing. This is really important. So the best way to think about this is to try and imagine what's happening. So at inspiration, what is happening? So inspiration, air moving in to the body. Expiration means air moving out of the body. Simple, right? Simple. Kind of makes sense. So air's moving in. Now, let's talk about our knowledge of diffusion and pressure gradients. So we know that substances like moving from high to low pressure. That's the idea. These are your basic principles for these two processes. So if air is moving in, remember, I can't change the pressure of the air because it's just the surrounding pressure. But what I can do is I can change the pressure internally to make it easier for air to either move in or out. So if we're saying inspiration is air moving in, what we're doing is we're moving the air, we're moving from a high pressure to a low pressure, which means inside our bodies, what we're trying to do is we're trying to lower the pressure. The best way to lower pressure, the third principle, that will be the baseline of every piece of knowledge, the lower the pressure is due to the higher the volume. Remember that? Kind of logical. And we do remember that kind of principle. So 3 principles that you need to remember. And these are principles of biology. So if you've got the principles of biology down, you're sorted. So high to low pressure, key. And we create low pressures by increasing volume. And inspiration is air moving in. Expiration is air moving out, or exhalation. So let's think about that. So inhalation, feel it. So whenever you're in an exam and you're confused about which order this goes in, do it on yourself. So you see, you breathe in, and you can feel your chest expanding. The idea is that your volume in your chest is going to expand. And the way this happens is you can feel the muscles in your intercostals contract, and you can feel your diaphragm contract because your diaphragm has to contract in order to move down. So the diaphragm contracts, moves down, your intercostals contract and bring your rib cage up and out. That's kind of its function. It's bringing your rib cage up and out. This means that overall, the volume in the thoracic cavity is going to increase. If the volume in the thoracic cavity increases, the pressure decreases, and the pressure then becomes lower than the pressure of the surrounding air. Air is drawn into the lungs. Simple. Exhalation is the opposite of that. So if you feel it, everything's moving back. You can watch it on me. You can visibly see the chest contract and relax. And so exhalation, both the diaphragm and intercostal muscles relax. The chest cavity contracts, which increases the air pressure in the chest cavity. There is a decrease in thoracic volume and an increase in thoracic pressure above atmospheric pressure. Air moves out of the lungs. That kind of all follows our basic principles. Why does the chest cavity contract? The chest cavity contracts, meaning not that that doesn't mean that it actually contracts. What it means is the size of it contracts, which means it gets smaller. Contraction in the other term for it, we'll change the term. The chest cavity decreases in volume. It gets smaller. It doesn't actually contract. Okay, so now we're talking about the efficiency of gas exchange. And we're now going to talk about a little bit of medicine, which is quite interesting. But the efficiency of gas exchange can be measured through a process called spirometry. And spirometry is basically a technique of measuring the volume of air exchanged between the lungs and the atmosphere. Now, this can be either normal or it can be abnormal because you have a different number of conditions. Different number of conditions affect the lungs in different ways. And spirometry is a really important diagnostic tool for measuring the normal activities of the lungs and for telling you if there are any problems with the normal activities of the lungs. And it is really important in the diagnosis of many diseases like asthma, chronic obstructive pulmonary disease, IPF, pulmonary fibrosis, PE, loads and loads of other conditions where things go wrong with the lungs. Usually spirometry at some point is involved in diagnosis. This is a really important table to take down, but it's only as important as understanding it on the graph, which is why, as much as I'm going to expect you to take this table down of some description, I'm going to give you a second or minute or two to write this down. I want you to imagine this through the graph. You need to think about it through the impression of the graph. So I will turn off my video, give you some time to write this down, and then I will talk you through the graph. Give you until maybe 35 past to write this down. Okay. Got a couple of questions. For the residual volume, is it talking about air removed from the alveoli? No, it's talking about the air left over with the alveoli once the alveoli is finished contracting. Because again, remember, this is about pressure gradients. Pressure gradients, at some point, the pressure is going to equalize. At that point, there's going to be some volume left over. Mariam, Do you know what a doctor is measuring when they're listening to your heart rate and they ask you to fully breathe in and out? Yeah, they're listening to kind of how your bronchi are functioning. So basically, if I'm doing this and I'm sitting here with my respiratory exam and I'm listening to it, what I'm measuring for is, do you have any wheeze? Or is there any sort of, how does it sound? It should sound like normal airflow, vesicular. And if it doesn't, that usually indicates something has gone wrong in this process. What factors will affect someone's vital capacity? That's a good question. Lots and lots of factors. So your vital capacity is if there's resistance in this process. So basically, anything that can cause resistance to this process. A couple of things that can do that. So COPD, so chronic obstructive pulmonary disease, which is a condition that affects people who tend to smoke more. Basically, two things that happens with that: chronic bronchitis, which is the bronchia become inflamed. They become inflamed. They often have a lot of scarring tissue, fibrotic tissue, so fibrous tissue can cause resistance to airflow. But they also have something called emphysema. Emphysema is the damage to the alveoli. And when you've damaged the alveoli, you can't get as much air in, which means you can't get as much air out. Other things, asthma. So asthmatic patients, if they're having an asthma attack, they can often have a lot of mucus that's built up in their airways. And if that's the case, then there's resistance to flow, which will affect their vital capacity. So again, lots and lots of factors can affect someone's vital capacity. I've been asked a question: why will there be some air left over? So let me imagine this. So remember that everything occurs on a pressure gradient, right? So the pressure of the external environment is going to stay constant. As you're breathing out, your low pressure, as you're breathing out, your pressure goes from high, but it slowly gets lower, right? But it doesn't go back below this, because at this point everything is relaxed. So this doesn't really happen. But it reaches this point. And it reaches this point where there's still some air left over, where, because the pressure has reached an equilibrium, so not all of the air has gone out. And that is the residual volume. It's the same way, a parallel to draw to that is the same way we talked about tissue fluid last week. And we said that not all the fluid comes back in at the venous end. Some fluid is left over. That fluid is lymphatic system. That fluid is lymph fluid. Make sense? Yeah. Okay. Remember, breathing rate increases during exercise or physical activity, which means that there's an increase in the amount of air exchange and an increase in PVR. It's really important to know the graph, though. So your tidal volume, normal breathing. Your residual volume is basically the maximum point that you can get out from your expiratory reserve volume all the way down to the bottom. Breathing rate and ventilation rate is the same. And then you've got your different capacities that are here. And again, the best way to learn this is to memorize it. Again, it's something you need to know from memory, but link it back to your definitions and you'll be fine. So I'd give you a couple of seconds to write this out and draw this out. Give it till maybe 41 or 42 past to draw this out. Again, very important to note for basically all exam boards. So here's something that will come up in most exam boards. So don't kind of neglect this content. It's very important content to learn. Could I please give an example? Chloe's asked, could I please give an example of what type of question would be asked about this graph? There's often, so some questions that could be asked, they could ask you to label some of the terms on this graph. So they could give you this graph empty, ask you to label it. Another couple of questions they could ask you, they could ask about they could just ask you a question about changes in the spirometry graph with certain conditions, especially for AQA, where you do have to have a little bit more condition-based knowledge. And in OCR, I remember there being a couple of questions where they're not going to ask you too much depth on this graph. They're not going to expect you to know about FEV1s and FVCs and things like that. You don't need to worry about all of those things. What you basically just need to think about is you need to think about kind of the different definitions of these terms. What they could just ask you is they could ask you, what does vital capacity mean? And you would just be expected to write down an answer for vital capacity. But again, it could be anything from definitions to labeling to explain. Sometimes they'll ask you to explain what's going on at the different points of this graph. And probably the hardest type of question will be something in AQA where they'll ask you to explain how this graph might change with conditions. What's meant by vital capacity? So vital capacity is your total voluntary breathing capacity, basically. It's the amount of air that can be breathed in and out so it's the difference between the lowest point that you can breathe out. It's basically, if I took in a very big breath in and then breathed all of that out, that would be my vital capacity. The change in that air volume would be my vital capacity. That's kind of the idea. Okay, pathologies. So pathologies are probably, if you know the pathologies are important for your spec, I know these aren't important for OCR-A particularly, but I am still going to go through them. They're quite interesting. If some of you do want to become medical students eventually, you might want to learn about some of these because these are quite interesting and important. But these pathologies are important for other examples. So asthma is basically an allergy, and an allergy leading to airway obstruction. Basically, what happens is you have an external trigger that causes you to have an allergic anaphylactic response. Basically, you bronchoconstrict. You bronchoconstrict and you inflame your airways. Once that happens over a long period of time, lots and lots of bronchoconstriction, many, many years of it, will lead to scarring and fibrosis of your airways. That will make it just harder to breathe in general, which is why chronic asthma needs to be prevented and you need to widen your airways. But bronchoconstriction, the idea is that it leads to a reduction in normal ventilation and deprives the body of oxygen, which is why asthma attacks, especially if they're severe and acute, they can be fatal. Again, asthma attacks severely can be treated with adrenaline. Adrenaline is a bronchodilator. Otherwise, for long-term care, use steroids or different other drugs like inhalers. You've got your blue and your brown inhaler that can be used. And again, steroids decrease inflammation. That's kind of their job. So you've got your normal airway. When you've got this allergic response, you produce lots and lots of mucus so that you're not getting the allergen into your lungs. You tighten you bronchoconstrict, you narrow the airways, and the idea is basically to stop whatever the allergen is, which might not even be a serious allergen, from getting into your lungs. The issue is that overreacting on this can then lead to more damage to your body, which is an ironic sort of chicken and egg situation about what's causing the harm, whether it's the actual allergen or whether it's your body's overproduction of this. I've been asked a question, how do inhalers help with asthma? So inhalers themselves don't inherently help. It's the medication that they contain. So you have loads and loads of different types of inhalers. You have your blue inhalers, which is salbutamol. Salbutamol is a beta agonist. So what it does is basically binds to a particular receptor, and that particular receptor is found on your airways. And then that receptor would normally be bound by adrenaline. So it's basically kind of replaces the adrenaline function on those receptors, and it causes the airways to dilate. The other kind of steroids work by actually affecting the cells themselves. So they work on directly targeting your inflammatory cells. So your white blood cells that normally cause inflammation, and they basically work by kind of dampening that response. You've got other inhalers. So you've got montelukast, which is a leukotriene antagonist, but again, that works on a different pathway. But the whole goal of these inhalers is to widen your airways again. That's kind of the idea of them. Are steroids a branch of lipids? Yes, so they're a subset of lipids. They're basically, they're lipophilic. They're also a type of hormone group. So steroid hormones like insulin are very common. They're a separate group of hormones. Okay, smoking and emphysema. So emphysema is one part of a clinical syndrome called COPD, chronic obstructive pulmonary disease. And COPD consists of two main syndromes. The two main clinical syndromes are chronic bronchitis, which is basically a long-term inflammation of the bronchi, and emphysema. So emphysema is the destruction of lung parenchyma, which is lung tissue, and it basically is the destruction of the alveoli specifically. It does that by destroying the septae between, so the splits between the alveoli. It also does that by destroying the tissue around the alveoli, which means that alveoli can't expand, stretch or recoil, which means that which means it's not as efficient for normal gas exchange. And emphysema does this by breaking down a molecule called elastin. Elastin is your elastic fibers that exist within your alveoli. Is this on the AQA spec? Yes, this is. The reduction in normal gas exchange reduces the ability to do physical activity because you can't get as much oxygen into your blood, which means you can't get as much oxygen to your cells, which means you can't get as much oxygen around the body. Smoking and airway disease. Chemicals in cigarette smoke, i.e. tar, can severely damage or obstruct airways. Smoking can cause airway inflammation, and cigarette smoke can also directly damage the ciliated epithelial cells, leading to an accumulation of debris in the airways. This causes an individual to cough excessively. So there is a lot of damage that smoking can do to your body, which is why, it's recommended that whenever a patient comes in who has got a history of smoking and is displaying symptoms like coughing, they're struggling to breathe, they might not be able to they're not able to do exercise as much. They might be, feeling quite ill, feeling quite tired. These symptoms, you have to advise them to stop smoking, because stopping smoking can directly impact the individual ability to work properly. And inflammation is a common part of different diseases, so chronic bronchitis and chronic obstructive pulmonary disease as well. Smoking is also heavily linked with cancer. So smoking contains many carcinogens, which can directly cause cancer. And again, the main ones are lung and oral / esophageal cancer. Esophageal cancer is also linked with high alcohol intake. And again, very important to know. The last condition that you kind of want to know is something called pulmonary fibrosis. So pulmonary fibrosis is one of many diseases that are considered interstitial lung diseases. Basically, between the alveoli and the capillaries, there's something called the interstitium. The interstitium is this space that exists between the alveoli and the capillaries. And in pulmonary fibrosis, the walls of this interstitial space, so the interstitial space gets filled in with loads and loads of inflammatory diseases, because these inflammatory diseases think that there's some sort of damage going on to the walls of your alveoli when there isn't. So they're overreacting to damage that isn't really there. And fibrosis is an aberrant or an abnormal repair pathway. And when you have lots and lots of activation of pathway, of this repair pathway, your body goes, I can't keep up with this. I literally cannot keep doing this, keep repairing this. I don't have time for anything else. So what we're going to do is we're going to put in together lots and lots of fibrous tissue. We're just going to keep it there, going to hold it, and we're going to try and maintain that. That's it. Because it's much easier, much quicker than going through the proper repair pathway that would normally happen if you've got an injury. Basically, so fibrosis is a replacement and repair pathway. And again, fibrosis is, remember, it's fibrous rather than elastic tissue. Fibrous tissue is tough. It's not flexible, which basically means your alveoli can't stretch and recoil as easily. Your alveoli then lose their ability to kind of take in as much air. And so you have reductions in your tidal volume, your forced expiratory volume, and your forced vital capacity. And again, it thickens the walls because it's thick fibrous tissue. And again, that means that there's less efficient gas exchange. Anaphylaxis, let's see, that's the last condition that we need to talk about. Anaphylaxis is an extreme allergic reaction resulting in airway obstruction. So asthma can cause anaphylaxis. And again, anaphylactic shock, I don't know if anyone, hopefully no one has experienced it, but it is a serious kind of a medical emergency. And you often see people with chronic allergies carrying around EpiPens, epi for epinephrine or adrenaline. Adrenaline is a bronchodilator. The idea is you inject them with adrenaline right into their muscles, right into their bloodstream as quickly as you possibly can. You bronchodilate because anaphylactic shock can kill you within minutes. And it's a serious cause of both mortality and medical damage in the long term, because the longer it is that your body goes without oxygen, the more your cells just start to die. So very important that that we understand kind of using kind of EpiPens, and everyone should use EpiPens correctly. But yeah, anaphylaxis is a chronic, severe, it's an acute, sorry, not chronic, but it's a severe allergic reaction to an external stimulus. Again, anaphylaxis doesn't need to be, so asthma is caused by inhaling your allergen. So usually an asthma allergen is like cold air or like pet hairs. So it's something that you can inhale. Anaphylaxis can even be caused by eating the allergen, or in other ways of getting that into your body, because anaphylaxis is systemic. It's a systemic allergic response, whereas asthma is fairly localized. And again, anaphylaxis is mediated by a molecule called histamine. Histamine is produced by your white blood cells, which is why in long-term management of allergens, you can often give steroids because steroids block your mast cells or your white blood cells from working, block them from releasing histamine, so you don't have the anaphylactic response. Bronchodilators, so epinephrine, which is found in EpiPens, which you inject into your thigh. But yeah. Okay, so last bit is talking about lung disease and pollution. So pollution has similar effects to smoking, probably at a lighter concentration because it's not directly going into your lungs. You're not sitting there and breathing the chimney of a factory. And so it's probably not as severe. It's probably not as localized. But pollutants in the air, i.e. nitrogen oxides, dust, and debris, can damage lungs, can cause emphysema, chronic bronchitis, and lung cancer. And there was a really interesting study done a couple of years ago, and this is just external knowledge, but they were looking at people who'd grown up in cities versus people who'd grown up in the countryside, and they found that actually people who'd grown up in the cities have significantly worse lung capacity than people who grew up in the countryside, and it was probably because of the availability and the kind of environmental impact of pollutants. Whenever you're looking at questions about data application, and often you will get data application questions, I'm specifically looking at AQA students for this. This comes up often. AQA students, you often get questions about pollutants and lung capacities and things like that. Don't be tricked by your units or your axes. Often they will be talking about thousands of people rather than individuals. So remember to look for that, and remember to look carefully at all of your axes and carefully at all your units. Remember, there might also be an axis break, so watch for that, because often it might not flow the same way you expect it to. It might start at a different point. So remember that as well. And your different types of questions. So when they're asking you to describe something, what they're asking you to do is they're asking you to basically tell them what's going on. So use figures, but tell them explicitly what's going on in this graph. Don't explain. Explaining is why is it going on? So why is this trend the trend that we see? And then discuss is, so you'd normally be discussing the validity of a particular kind of hypothesis, and you'd be given data, and what you need to do is talk about some pros, talk about some cons, and give me an evaluation on your overall thoughts. Remember, causation versus correlation, they're not the same. So causation is X causing Y. Correlation is where X and Y could have both changed in a certain way. That doesn't mean that they could be unrelated. So if we talk about since 2002, the rise in smoking has led to an increase in the number of patients suffering heart attacks. Since 2002, there's been a rise in smokers and the number of patients suffering heart attacks. Basically, the idea is that the smoking might be a correlative. It might have an effect on heart attack rate, but it's not going to cause it directly. Smoking is an act. The way I think about that, causation basically means everyone with that risk factor should get that condition. For example, everyone with a mutation in a particular gene, I'm not going to quote genes at you, but everyone in a mutation in a particular gene will eventually develop Huntington's disease. That means that the mutation in that gene causes Huntington's disease. Now, if not everyone with the mutation in that gene, or if there is a factor that not everyone with that factor gets the condition, then it's a correlative effect. It's not causative. So, for example, if obesity and type 2 diabetes, so not every obese person will develop type 2 diabetes, but obesity is linked to type 2 diabetes. So obesity doesn't cause type 2 diabetes. Insulin desensitivity causes type 2 diabetes. But obesity can correlate and increase your chances of getting that condition. That's kind of important to know. We're not going to really talk about those, but that's kind of it. I know today was quite a long session. Again, we only focused on the human aspects of it. I would have liked to focus on some of the other insects and fish, but we did kind of cover quite a lot of the human aspects of it. Do we have any questions? Oh, I've asked a question. Do your alveoli ever go back to normal if you stop smoking? No, alveoli don't recover. So that's better not to smoke at all, because alveoli aren't recovering. This recording will be on YouTube on the side. So and all the previous recordings are available. Also, I was wondering if I was a medical student. Yep, I am a medical student. I'm a third year.